All right, our next presentation is by Steve Dent, PhD. Steve has a bachelor's and master's in environmental engineering from Montana Tech and a doctoral degree in civil engineering from Washington State University. The bulk of his graduate work involved investigating the effects of lake and reservoir treatments on the fate and transport of mercury in the aquatic ecosystem and maintaining a working trace mercury lab. Over the past 10 years, Steve has developed and applied investigation strategies at mercury-contaminated sites all around the U.S., with particular focus on mine sites. He's currently the mercury subject matter expert for CDM Smith, and he's going to share his presentation on investigation strategies for investigating fate and transport of mercury at mine sites. So, welcome.
Thank you, Steve. Well, thanks for having me here. I'm here to talk about as Steve mentioned, investigation strategies for evaluating fate and transport of mercury mine sites. And it's not really a comprehensive overview of all fate and transport for mercury. It's essentially a review of mercury cycle and some of the unique aspects of fate and transport evaluations because mercury is a very unique metal. And it doesn't have, it's got some unique properties that other metals at contaminated sites don't have, and that provides challenges. It also provides opportunities for effective investigations, which haven't always been very successful historically in our field. There's a lot of sites that have thought they've buttoned it up and walked away, and then it turns out 10 years later it's still a problem. This is also a kind of precursor to next year's talk. We have a data set that's not quite ready to share with everybody yet, but we'll be debuting that this summer. And so next year I'm hoping to be able to come here and share our results from one of our sites. I think it's pretty exciting. So first thing I'll do is give you an overview of risk drivers, which are a little bit different for mercury than other metals, and then talk about the types of mines where we see mercury problems most often, and then a little overview of how we look at the conceptual site model of mercury at contaminated sites, specifically mines. And then just some considerations that are unique from other metals on how to investigate mercury, and then a quick summary. So to start off with risk drivers, a lot of contaminated sites with metals issues, it's about exposure, and it can be about exposure for mercury at the mine site itself, where the point source is. But oftentimes the driver is downstream, whether it's in the river or the final receiving water body. And because of that, we deal with concentrations that are pretty low, much lower than any kind of exposure risk. And here's a summary of mercury advisories based on consumption of fish in the United States. And you'll notice there's two colors here on each state. Blue represents states that have a blanket statewide consumption advisory, where they've gone out and they've not been able to really come up with any metric that they can identify a lake that would be either above or below the consumption guideline, and so they just put a blanket on the whole state. Other states in yellow are site specific, so they've gone out and they're doing their due diligence in sampling each of these water bodies and saying whether or not they are under a consumption advisory, which gives anglers an opportunity to identify a place to go and catch fish and eat it without concern for at least mercury. So when we talk about mercury in mining, the most kind of obvious one is mercury mines, right? Extraction of elemental mercury from some sort of mercury ore, typically cinnabar. And that's a process where they go, use traditional methods to extract mercury ore and crush it up. And what they do is, it's called pyrometallurgical extraction, where they take crushed up ore and roast it in a kiln. And then that creates a conversion with oxygen, mercuric sulfide, which is the composition of cinnabar, with oxygen in heat, generate elemental mercury and sulfur dioxide. And because elemental mercury is a liquid, it's the only metal that is a liquid, and it volatilizes, and just like you're making hooch or distilling liquor, right? You run it through a condenser and bring the temperature down, and it turns back to a liquid, and they collect that. And back in the day, they did that quite a bit for gold and silver mining, which leads me to gold and silver mining. That's another type of mine processing site that you will see mercury contamination. And these sites aren't necessarily geogenic sources. They are gold and silver deposits that are mined, but they brought mercury in to help extract the gold and silver. This is a map of California. All of the yellow dots here in California are old gold mine sites. Almost every single one of them is contaminated with mercury because they brought mercury in. And they didn't really have that great of safe handling methods back then. You'll also notice a handful of these red dots a little bit further west in California. And that's a band of, altered zone where there's a lot of mercury deposits. And so what they would do is try to find mercury deposits near all these gold deposits, extract the mercury to then help isolate gold. And for silver, it's the same process. You have it if you have an old silver, filling in your tooth. It's an amalgam. It forms just like, gold and mercury form an amalgam. There's a bunch of mills around Butte that have mercury hotspots around them because they brought mercury in to extract silver from silver mines in Butte. There's one near Peaburg. So common processes back in the day when it was legal to use mercury to extract gold and silver. So when we talk about the mercury cycle, here's a big, giant, messy kind of global view of how mercury interacts with the environment. Over here, we have, your mining activity. You have your coal-fired power plants. You have other industrial activity. Coal-fired power plants and mines often release mercury to the atmosphere. Mercury is a transcontinental pollutant. It circumnavigates the globe. And with atmospheric processes, it can oxidize and then deposit over the landscape. And there's not 1 square foot on Earth that doesn't undergo at least some portion of mercury deposition. That eventually makes its way down through rivers and into your water bodies, where it undergoes other transformation processes that I'll talk about in a second and builds up into fish. So to simplify that down a little bit, we're talking about inorganic mercury, Hg2+, converted into methyl mercury. It's called mercury methylation. It's facilitated by anaerobic bacteria, the primary offender being sulfate-reducing bacteria. And it needs to be in an anaerobic zone to work, right? And so since these are anaerobes, they operate when oxygen and nitrate are all consumed, but they need to be fairly close to an oxic zone because they need fresh oxidized substrate. Sulfate-reducing bacteria needs sulfate to operate. So it needs to be right close to the surface of that redoxycline. And so that kind of helps it enter the surface water because it usually generates in sediment pretty close to the actual sediment water interface. Other factors that are required for it include different types of carbon. The type of carbon, the quality, and the amount also impact mercury methylation. Some forms of carbon combined with inorganic mercury and prevent it from being methylated. So it makes it unbioavailable. Other forms of carbon, the sulfate-reducing bacteria can use it as a carbon source and bring it in along with mercury. And so when we talk about mercury methylation, whether it be in pore water, and it has to be in the aqueous phase to become methylated. We talk about it in pore water, surface water. You can have methylation in surface water. And we also have this new up-and-coming category here, periphyton, attached algae in stream channels that actually can help facilitate mercury methylation outside of the sediment. But typically what happens, you have inorganic mercury. It can be methylated into methyl mercury, or it can be reduced into elemental mercury. Elemental mercury can be oxidized into inorganic mercury. You can have demethylation occur. So there's microorganisms that facilitate demethylation, which cleave the methyl group off. There's also photodemethylation here, where sunlight cleaves the methyl group and reduces it to elemental mercury, and then actually can evade the system. Here's a simple cross-section of a stream, just showing kind of some of the processes at work here. Atmospheric deposition directly deposited to the surface. We have riverbank soil erosion occurring, right? You have sorption, desorption from sediment surfaces. You have sedimentation and resuspension, right? So generating secondary sources and then resuspending them and sending them downstream. You have soft depositional sediments as a potential player in here, and we also have like a cobblestone bottom channel that can be a player. In the depositional sediments, that's typically where, in the channel substrate, you're going to find mercury methylation. In the cobbly substrate, you have hyporheic flow under the surface, but not necessarily groundwater, but not necessarily surface water. But we find a lot of dissolved mercury being transported down in these kind of under surface layer zones. When we talk about methylmercury, it either enters the environment by in situ production. So in an aquatic watershed, you have mercury methylation that happens within your zone that's impacted by some sort of point source from a mine. But you can also have methylmercury transported from other areas, right? Upstream, maybe there's a wetland alongside, or wherever those other sources might be. So it's generating in situ, and then you have other sources entering. And if you talk about a reservoir, what you're talking about is methylmercury in your stream entering the reservoir. The reservoirs are usually a hotbed of mercury methylation because of all the soft sediment in the bottom, anoxic bacteria, anaerobic bacteria going crazy in the sediments. But you also have a load of already methylated mercury entering the reservoir that can help impact food web uptake. And so, but methylmercury itself isn't very soluble, so there's a lot of ligand binding going on, and the type of DOC and TOC in your system is going to affect how it moves through the system, right? So if you have TOC that has a propensity to bind methylmercury, it can stay locked up in sediments. Penobscot River up in Maine has fairly low methylmercury in sediment, but pretty high uptake in the food web because the sediments, the character of TOC in the sediments aren't conducive to lock in that methylmercury. So it actually just kind of releases everything as it's being methylated. And so that helps exacerbate the problem there. Aromatic DOC is a type of carbon from autochthonous sources up in the watershed where it has a really strong propensity to bind with mercury, especially methylmercury, but it can actually outcompete the food web for methylmercury because typically methylmercury enters algae in the water, but if you have aromatic DOC in your water, that can actually prevent it from building up into your system. So this is what makes mercury so complicated. It's so very site-specific on all sorts of parameters. So there's this new paradigm, periphyton in the aquatic ecosystem, right? So periphyton is attached algae that can grow on cobble substrates, rocks, bedrock, if you have any bedrock in your system. And what that does, it's a photosynthetic amalgamation of photosynthetic algae, bacteria, fungus, dead plant matter, sediment. And what we're finding is it has a capability of methylating mercury. And it's another source, another rock to turn over when we're looking for sources. There's a lot of sites, especially rivers, where we're having difficulty finding where the methylmercury is coming from because we always just look at sediments. And now we're starting to find that we need to start looking at, substrates like periphyton. Okay, so methylmercury bioaccumulation, it's an interesting topic over the last 20 years talking to people about the use and utility of trace mercury analysis, right? We talk about concentrations in the 1 nanogram per liter, and I've had to, try to talk people into that. It not only is it real, it has utility. I've talked to folks in the industry that it's not a real value, or why would we care? And this is why you care. Let's take a top-end food prey fish or predator fish, right? So our consumption guideline from the EPA is 300 parts per billion, right? So that's where we're going to start. So the general model is a 2 to 5 times increase each stage of the trophic level as mercury passes up, as it's being consumed. So let's work backwards. So we have, prey fish here, 60 to 150 ppb. We have, zooplankton, benthic macroinvertebrates, 12 to 75 ppb. Algae, 2.5 to 38 ppb. And then the big jump from water to periphyton, or water to algae. And that leaves us with a starting concentration of methylmercury in surface water at 0.024 nanograms per liter to 0.375 nanograms per liter, right? That's a very low concentration, but that is a range that could potentially get you to that 300 parts per billion in predator fish. And because of this, the California Control Board, which, that's the state that has to deal with most mercury issues because of the gold rush, they're setting a standard for surface water of 0.06 nanograms per liter in their TMDLs. And that's because they realize what concentrations they have to start targeting for methyl mercury. We're not talking total here, but methyl mercury, because they have such a huge problem in their reservoirs, because almost all of their drinking water reservoirs are in the watershed of an old gold mine. And so the general model here is from water to human and ecological receptors, just to kind of show you in a nice, neat package. All right, so let's talk about some unique concerns here related to fate and transport evaluations. So here is a typical setup of a mine in a watershed with a reservoir downstream, right? And so there's several things that can happen. You have your source area here, you have tailings piles, and it's draining into a nearby creek or river, right? And as it moves down, this could either just be a conveyor belt that, there's not much activity at all in the system until it gets to the reservoir and then starts methylating, or there could be all sorts of processes along the way that, create secondary sources and also mercury methylation zones. And so sometimes when you go to sites like this and you start evaluating the food web, you start seeing a massive spike right below the mine in your anywhere from your benthic macroinvertebrates up to fish. And you see subsequently high levels in the reservoir as well. But something usually has to be happening in the actual river system if you're going to see elevated mercury above background in your flowing water. So what you do is you start looking at your sources, right? So primary sources, you have elemental mercury, right? Both mercury mines, gold mines, and silver mines have elemental mercury associated with their contamination, right? Because you're extracting elemental mercury at the mercury mine, you're using elemental mercury to extract gold at the others. Mineral-bound mercury tends to be more associated just with mercury mines, right? Because you're digging it out of the ground, crushing it up. And it's pretty heavily mineralized, not very mobile. And elemental mercury isn't really mobile as well. But then there's also the inorganic mercury, which the tailings piles, the calcines, after you've done roasting them, has a lot of very labile mercury salts, inorganic mercury that can mobilize. And then over time, elemental mercury oxidizes and becomes part of that transportable pool of inorganic mercury. Now, you talk downstream of the mine, you have secondary sources, areas where mercury accumulates and it can potentially be re-entrained into the water column. Now, close to the mine, you're going to have a pretty similar suite of constituents right below the mine in that drainage. But as you move downstream, it starts to become dominated by inorganic mercury because inorganic mercury is the mobile form. But when you get to background sources, this is what makes mercury really tough, is when we talk about background characterization. You have atmospheric deposition occurring on your watershed, right? It's dependent on the region because it changes depending on where you are, right? If you're in the tip of the Northwest, you're going to have extra high mercury deposition because you're closest to China, and we start getting deposition of atmospheric mercury from China. We see it correlating with their ramp up of their coal-fired power plants. Homeland Security guide, that might be a risk there too. Geogenic sources, right? If you're in a mercury mine area, the geology around that area is pretty elevated compared to other areas. So your background sources could potentially be in just a naturally heightened concentration of mercury as well. Then there's your mine influence sources, and I say that because we have a site that anecdotally, we haven't confirmed this, but the locals, because this particular mine, we shut down in the 60s, the locals, oh yeah, my grandpa told me that they took the mine tailings and the logging company built roads with them way up in the hills here, and that's in our background area. So there's a potential mine influence that's impacting our background area. And then there's other anthropogenic sources, right? And that's like with any kind of contamination, right? So you have a Superfund site, you have to deal specifically with sources from the Superfund site, and there's some sort of dump up in your watershed above that that's contributing mercury, but you're not there to solve that problem. You have to differentiate it from your source, and differentiating it is the challenge. And so in comes isotopic fingerprinting. So just to kind of give you a chemistry 101 overview here. What makes mercury mercury is 80 protons, and when it's neutral, 80 electrons, right? That makes mercury mercury. Now the neutrons can vary. You can add neutrons, you can take neutrons away. It's not going to change the charge balance. It's just going to change the mass. And that's why we have a different set of fractions of different isotopes. And these are the weights of these different isotopes. And the size of the pi wedge is their relative contribution to the suite of isotopes. When you collect a sample, it generally follows this distribution of isotopes within a sample. Now we have a national standard that we can use that's set in stone. All these pi wedges, all that the percent from each one is locked and set in stone. And so when we take an unknown sample, we can send it in and have it characterized. And the difference, the change between different isotopes as compared to the national standard gives that sample its individual fingerprint, because every source has just a little bit different fraction. Atmospheric deposition has its own mix in fingerprint in region to region. And so when you do that and you kind of look at background sources, your mix zone, and your actual tailings contribution, and you plot this out with differences in, like, say, for example, mercury 199 and the changes in mercury 202, you have the red here, which is your mine signature. You have your blue here, which is your mix zone downstream. And green here is your background samples. And this is an example from one particular site where, we're trying to figure out where to collect background. You can see how distinct each of the background areas are from each other. So selecting the proper background is important, but what we can see here, there is definitely a distinction, and it all kind of makes sense. This is kind of a mix between background and tailings. And what we can do with that is we can try to use a mixing model. And to use a mixing model, we need to pick background sources. So here is a mine site here outlined in black. This red stipple is your kind of altered zone, which has high mercury mineral content, and so that's our source area. Then we look at our background areas. You can pick 2. You can pick more than that. You can mass balance and put them all together and make a mixture to compare it with of background. And then what you do is you try to you use these as end members. And then you grab this mixed sample at the bottom, and what you do is you can, using a mixing model, get a percent contribution from those tailings. And if you plot them out, it can look like something like this. Per river mile, moving downstream from the mine, we can kind of average it out. It's about 55% mine contribution, all the way to 11 miles down the stream. Right? What that does is gives us a potential target. When we're trying to figure out what do we got to clean it to? What's the mine contribution in all this sediment? Well, that's one way to do it is, and you can do that with the reservoir. You can do that with, as far down the stream as you want. The challenge is when you have multiple sources. If you just have one ore body to work with, that's fine. In California, they have like the San Francisco Bay, which is impacted by, 6 different ore bodies. And they're trying to differentiate whose ore body is it from, right? And that gets really complicated. But when you have just one mine signature, it's starting to evolve into a point where we can potentially someday use this to develop our PRGs, for example, our remediation goals. And it's a pretty handy tool. Right now, we kind of use it as a little line of evidence because it's still fairly new. So when we talk about mercury entering the food web, right, methyl mercury starts entering the food web in the surface water. So what we also have to do is try to figure out, we have transport issues, we got the source issues. Well, where is mercury methylation happening? So when you come to a site, the typical mercury methylation zones, depending on what's at the site, it can be depositional sediments. That's a pretty standard place to look. Saturated soils, depending on the site. If you have soils that are constantly saturated and have a lot of organics in it, that's a potential methylation zone. And wetlands are a big one. If there's wetlands along the corridor at all, that can be a major source of mercury methylation. Less typical, but still relevant, Paraphyton, there's some studies over the years that had shown that, and some sites of the Oak Ridge National Labs is starting to find paraphyton as a mercury methylation zone. Suspended flock, just kind of dense flock suspended in the water column can potentially methylate mercury. And then in some water columns, it can methylate within the water column itself, more typically in like deeper lakes and reservoirs. But with that, I want to tell a story. I think I have just a little bit of time. We went to a site and we did our typical, like, here's our site. We're going to look at sediment, pore water, surface water, and fish, right? So we look at those constituents, and we see a good amount of mercury in fish. We see methylation happening in sediments, and it's slightly elevated in surface water. So if we had stuck to it there with our budget the way it was and walked away thinking we had it solved, we would have walked away and not solved it at all. But we fortunately had a teaming partner that went out and did a bunch of sampling on benthic macroinvertebrates. And they happened to, just by chance, include snails. And those snails came back so hot that the people sampling it said this was they do regular mercury sampling at sites all over the place. And they said these are the hottest snails they've ever seen. I don't think I've ever planned to say that in my life. These are hot snails. But so we went back out and we looked at the paraphyton, and the paraphyton was either fairly close or a lot higher than mercury methylation or methylmercury in the paraphyton than in the sediment and pore water. And so it was a kind of a aha moment. And the depositional sediment actually was a pretty small footprint in the overall watershed, but paraphyton was everywhere. It's that slippery slime, the biofilm on rocks, why so many fly fishermen and women put felt on the bottom of their boots so they don't slip. That's the biofilm. Then there's also the filamentous, right? Filamentous, long, wavy, like green hair flowing in the creek, right? Not all sites find it as a methylmercury zone, but here we found it very significantly high, and it was ubiquitous everywhere. And so I'm showing you here is some artificial substrate samplers we have out here in a shallow river, here in a deep river, where we've used these samplers to collect and evaluate paraphyton over time, also as a passive sampler. But next year, my plan is to come with the results from this. We're debuting it in South Africa, in Cape Town, at the International Mercury Conference in July this summer. And so with that, just to sum it up, fish consumption is typically the risk driver for mercury. And it's also the hardest risk driver to solve. Mercury, gold, silver mines are the primary suspects when it comes to mine sites that have mercury issues. It's a challenge because of the unique properties of mercury. The background is probably the biggest headache to try to nail down, is a good solid background for these sites, because it's kind of everywhere. You can have background contributions that cause fish issues without any anthropogenic activity in your system at all. And so you have to split those out. And also finding where methylmercury is occurring is also a challenge. But because of its properties, we have things like isotopic fingerprinting that is a new tool for the toolbox, and our growing understanding of how and where mercury methylation occurs is allowing us, more places to look and more effective investigations. So with that, I'll take any questions.
All right, questions for Steve? Okay.
So you talked a bit about the mercury methylation and transport resulting from that. What about mercury cyanide that comes from co-leaching of gold tailings or silver tailings, and the mobility of mercury cyanide? Does that change the fade and transport approach?
I haven't run into that. So the use of cyanide is kind of a replacement of I've not run into issues with mercuric sulfide or cyanide, so I don't know. I don't know if that's been a problem, but usually when it's a gold mine site, newer ones, there wouldn't be mercury associated with it.
Okay, more questions for Steve? All right. Well, thank you, Steve. It's a complex problem.